Good morning. I wanted to talk a little bit differently about this issue, more from a practitioner's point of view. The previous two speakers had fairly quantitative and fairly specific information about COI policies as well as what the impacts were. Mine will be more semi-qualitative, probably anecdotal, but I'll try to highlight some of the ways by which NCATS has been able to deal with conflicts of interest and thrive in the area of collaboration. So one of the things that was very critical when we got established in 2011 was the fact that the mission wanted us to develop innovative methods and technologies that will benefit the whole biomedical community. So as you can imagine, we had a fairly quick growth from 2011 through now. We have been able to put together a lot of different programs which are already in existence, but we formalized them. But one of the things we realized right from the beginning was the fact that we were given this moonshot problem with very few resources. So the only way we could solve the problems were through collaboration. So right from the beginning, we at NCATS have been clear in our mind that the best way to deal with it, and probably the only way to deal with some of the challenges that we have, would be through collaboration, because the problems are of massive scale. And in doing the exercise of setting the collaborations, we came with a number of different rules and suggestions from different parties. And I wanted to summarize those here in the form of some bullet points that might be useful when you think about collaborations. First and foremost, what we did was we wanted to set parameters for the collaboration in terms of clarity of responsibilities, clarity of the direction that we were going to be going, what the responsibilities were, what the goals were. And whenever we were looking at collaborative projects, we were making sure that there were synergies that were captured in the research plan or the development plan through the complementary assets that the different players are bringing in. Another thing that we wanted to do as a government agency was the fact that most people consider the fact that we would be an honest broker in terms of trying to ensure that different parties' interests are met in a very equitable fashion. So we started putting more and more of our voice forward as an honest broker and as somebody who would be transparent in the relationships between the collaborators. The other thing we realized in any biomedical research and development program is that not all projects are going to work out. So we decided right in the beginning that whenever it's important to disengage, do it gracefully and do it in a manner that preserves the relationships for future collaborations. So within the realm of collaboration, we realized right from the beginning that when you talk about collaboration, it has to go right from the place where you're starting. In other words, collaboration should start at the institution where you're working. So we made it a point that at NCATS, every individual, every group, every person that was working there had the collaborative DNA instilled in them. So all of our projects have multiple people leading them. For instance, any biological project will have a biologist and a chemist and a bioinformaticist who will be leading the project. So doing it in a collaborative sense. And in those groups, we wanted to ensure that anybody from the technician level all the way to the PI level were participants in the planning as well as the conduct of the projects. So they would be, for instance, part of the conference calls. They'd be part of the planning committees. They'd be part of preparing the slides for presentations. So we wanted to make sure that we were able to involve all the members of the teams in whatever functions that they were doing collaboratively. And of course, we wanted to expand that sphere and idea of collaboration within NIH. We pretty much collaborate with 10 to 15 of these different institutes at NIH. And we also made sure that outside of NIH, we had collaborators from a variety of different backgrounds: academia, nonprofits, biotechs, pharma. And in all these collaborations that we held with outside groups, we wanted to make sure that they were governed by a few important principles, that whenever we do collaborations with outside groups, we are not competing with them. that the goals are aligned, yet we are bringing complementary assets so that we are seen as part of a team rather than as competitors within the collaborative subclimate. The projects that we chose typically expand the pre-competitive space so that there isn't any sense of not having to share the glory, and it was a common goal of trying to move the pre-competitive space forward. The other thing that we did right from the beginning in terms of our approach with industry collaborations was to ensure that the scientists who actually formed the collaborative teams wrote the scientific research plans and drove the collaboration. So that actually made it very easy because scientists typically speak the same language, whether they're working in an academia or a biotech company or government or maybe a patient advocacy group. So when they speak the same common language, they're able to come up with common goals that can form a good collaborative glue. Most of the projects that we chose are on new technologies, so there will be a lot of excitement for new discoveries as well as new applications. The other thing that we did to make sure that we as the federal government was an honest broker was to ensure that the collaborations were typically selected via a solicitation process, an open solicitation process, so anybody could apply for it. And the projects were typically chosen based on peer review. So in all the selection committees, we would have members not only from NCATS, but also from outside. We had venture capital people, industry people, academia people, all weighing in on the appropriateness of the collaborative projects and the budgets that we were trying to put together. In all of these projects, we ensured that the NIH policies and rules and regulations were strictly enforced and communicated to the collaborators right up front. So in terms of trying to ensure that collaborative DNA, the collaborative climate, and the collaborative activities expanded within the framework of the conflict of interest, we ensured that we didn't ignore the COI, but we weren't paralyzed by it. We were immune to it, but we weren't completely debilitated by it. So we followed the rules, but we made sure that the rules followed the science. So we wanted to make sure that the science drives the projects. So in terms of project dissemination, project information, we disseminated it widely. We used objective review processes. Wherever possible, we standardized and streamlined the collaborative procedures. The other thing that we did right from the beginning was to make sure that the collaborations are formalized wherever possible. So it wasn't one of the scientists at NCATS collaborating with one of the scientists in industry. That may be how the relationship began, but we formalized the collaboration so that there was some sort of a collaboration agreement. It could be as simple as a CDA or an MTA or a CRADA or something. Depending on the nature of the collaboration, we had agreements which were either 1 paragraph in length or 10 pages in length. So depending on that, we made sure that we formalized the collaboration itself. And whenever possible, we tried to standardize the collaboration agreements so that we didn't have to keep reinventing the wheel. So I wanted to very quickly talk about 3 case studies where the idea of collaborations as a means to solve problems was made very clear. These case studies are from one of the groups that was started about 2 years ago. It's called the Trend, or the Therapeutics for Rare and Neglected Diseases. In that particular case, the therapeutic platforms had to be in rare diseases or neglected diseases, which are the typical definitions that you have for rare and neglected diseases. Typically, these are areas where most pharmaceutical or biotech companies would not venture in because the financial incentives are not very high. There is a fairly difficult valley of death to cross because there are a lot of good discoveries, but they don't go all the way through the development of the drug. In all these projects, what we did was we put together research groups and research plans that were incorporating all the members of the different parties. As an example, I'll give you an example of one of the projects. In the last 3 years, we've taken in 15 different projects. These are all different rare and neglected diseases. We've been able to go into the IND in 4 of them. And in every single project that we have taken under the TREND program, it's a very unique public-private partnership. As an example, there is a project that we are doing with Genzyme and the University of Cincinnati. In this particular case, this deals with a disease called pulmonary alveolar proteinosis, which is a lung disease that is a very rare disease. It afflicts less than 2,000 patients in the US. These patients have fluid accumulated in their lungs because the surfactant protein that gets used for air exchange doesn't get cleared. It gets accumulated, and protein fluids collect there and make it very hard for the people to breathe. And the current treatment is you essentially treat the lungs of these people as beakers. You put saline solution into them through intubation. Essentially, it's an in-hospital procedure. You shake them up, and then you drain the liquid. You do that 4 or 5 times, and you get the proteins out. It's obviously a very, very invasive, not very comfortable procedure. And you can see that, you can see that in these patient lung fluid, you can see going from a murky fluid to a clear fluid after a day at the hospital of a lot of hard work. So we thought there might be an alternative. There is a drug called GM-CSF, which is sold by Genzyme for a different reason. We thought this would be a good repurposing of the drug because this drug could potentially go into the lungs and stimulate macrophages there and have the macrophages eat up the lung surfactant and clear it by a natural process. So we have a 3-way collaboration between Genzyme, which is supplying the drug for free, Cincinnati, which has got the PI, who's got the expertise in the disease, and Trend, we have the wherewithals and the expertise to take this forward. So hopefully this will be one of the success stories we can report in a few years. The second program that I wanted to talk about was briefly described by Sally. So I don't need to talk about it in a great detail. But one thing that I wanted to mention was that this, again, is a collaboration between NIH, academic researchers, and industry partners. And we wanted to formalize it in a way that each project will serve as a stepping stone for the next project. So we don't have to keep rediscovering the relationship game again and again. The first part of the triangle, the grants, is pretty straightforward because we are giving money to the academics. We lay the rules down, and we are fairly transparent about them. And that is a fairly easy way to, easy problem to lick. The other 2 sides of the triangle are not so easy. When we went to different industry partners, the main concern was, if we give you these drugs, what is the guarantee that we'll get the IP back or we'll get, get the follow-on developments back? So what we did was we set up a memorandum of understanding with one collaborator, one of the industry collaborators, and as that gained traction, others fell in place. So we have now a total of 8 industry collaborators who are part of the program. The biggest challenge was to try and set up collaborative research agreement templates, which each academic institution and the pharmaceutical company could use. Eventually, we ended up doing 8 different CRAs, or collaborative research agreements, 8 for each of the 8 different industry partners, but which could be used by the different academics as a template. Just as a highlight of the program, the Memorandum of Understandings are in place now, and the CDAs and the collaborative research agreement templates are also available. We have chosen a number of programs to go forward. And just to give you an example of what happens when you have this kind of a crowdsourcing, when you have used or when you have drugs that are in the attic for which you're trying to find new purposes, if you provide them to these brilliant group of researchers, some of them think alike. And some of them say, yeah, they can all be used for this particular purpose, but then others don't. This particular researcher thought it'd be a good to put this particular mechanism of action to test. So we get a fairly wide variety of different projects. But the idea is to try and bring them all together in the same research collaboration template. So just to follow up on that, some of the projects had multiple applicants for the same compound for the same indication. Others had different indications that they wanted to follow. The last thing that I wanted to talk about in terms of a collaborative project that's of a fairly massive scale is something known as a tissue chip. So the idea is that whenever whenever a pharmaceutical company or a biotech company is trying to develop drugs, the main cause of failure in phase 2 and phase 3 is lack of efficacy and lack of safety. So toxicity and lack of efficacy tend to be major drivers of drug failures. And one of the things that we asked right from the beginning was, is there a simple or is there a straightforward way by which we could replace animals and humans with tissue chips? The idea would be to see if tissue chips can try and reduce the risk and reduce the cost of this development. And the idea is to try and have biological chips that are composed of the different, I know I need to complete quickly, but I'll speed up a little bit. But so each chip would have 10 different functional cells. The problem is massive because it's not only a biology problem, it's an engineering problem, it's a bioinformatics problem, it's a fluidics problem. And we realized that the only way to deal with this would be to go into collaboration. It is a fairly massive 3-way collaboration with FDA, not providing any funds, but providing a lot of talks and regulatory advice. DARPA providing about 75 million over 5 years, NIH providing about 70 million over 5 years, and hopefully getting a lot of academics involved in terms of these different RFAs that are available for the academics to apply. And as an example, there was a proof of concept where this group in Weiss Institute was able to take some lung tissue and show that the lung permeability can be abrogated by a drug. So I'll stop there and answer any questions during the discussion.